Hello students. In last class I have described about some properties of DNA. One of the property that I have described was the complementary versus incomplementary basis in the DNA. Mainly complementary basis I have described in my last class. Complementary basis kotha moi tomalokok kolo agor class to. Gotike eta second property of the DNA molecule. Zero DNA structure already tomalokok koa hoise. The second property, DNA second property, sense versus antisense strengths in the DNA molecule. Sense versus antisense strengths of the DNA molecule. Antisense strength or sense strength mane ki bujwa. This all I am describing because in early exams these are coming repeatedly in exams. So sense strength mane ki or antisense strength mane ki. Let me to again describe the early descriptions that I have made in the molecular base of inheritance in this chapter. The strand of the DNA molecule, out of two strands of the DNA molecule, you remind one thing, DNA is a genetic material in all organisms. DNA, bongkogotiyo padartho, living being, hokolo jibot, DNA whole genetic material. DNA is a genetic material in all living beings. So it primarily performs the function of transmission of the hereditary traits from one generation to next generation. Here DNA main কাম হল কি এটা জনুৰ পৰা বংশগতিৰ চৰিত্ৰসমূহ আন এটা জনুলৈ স্থানান্তৰ কৰা. Transmission of genetic traits from one generation to next generation. This is the primary function of the DNA molecule. Okay? Kintu as I have spoken, the DNA molecule is made by two strands. DNA dual strand thake. The two strands run anti-parallel to each other, and we represent a DNA molecule by this formulation. I mean, DNA molecule presentation to keneke dekho paru. This is the two. These are the two strands of the DNA molecule. One strand run in 5-3 direction, and the other strand will run in the 5-3 direction, but in opposite directions. 5-3 one, 5-3 in the other. 5-3 in that direction, and 5-3 in the other direction. Okay, as I have said, the sugar and phosphate bases in the DNA molecule are, they are always common for every DNA molecule. Potato DNA karone sugar phosphate eke thake, but we base represent koro. The DNA we represent by bases only. So suppose this is in this particular place, base A is present, and this is the other base, suppose T is present in some nearby and little right, and next base suppose it is Z present, again C present, again suppose A present, T present, Z present, and C present. These are so and so on. The DNA bases are arranged in the DNA molecule, bases are arranged in DNA molecule. Okay, according to complementary base pairing rule, if in one strand of the DNA molecule A is present, in the other strand there will be T. Jodi adal strand of A thake, andal strand of T thake. And so also, if in one strand there is T, the other strand will contain A. If in one strand there is Z, the other strand will contain C. If there is C in one strand, there will be Z in the other, and so on and so forth. But A hole T, T hole A, Z hole C, C hole 0. This is exactly the presentation of a DNA molecule. A part of the DNA molecule I am representing here. That DNA part molecule a part of the DNA molecule I am showing here. Out of these two strands of the DNA molecule, while DNA is working, DNA come koribole jai. When the DNA functions, you remind one thing. That DNA acts as genetic material and transmits the parental traits from one generation to next generation. DNA parental trait black at a generation carry kore. Kintu carry koribole jaote DNA RNA dara kam kore. DNA works by producing RNA. Tumar rakhiba. In case of human being or other higher organisms, the function of DNA is genetic material, but RNA cannot act as genetic material there. Mane jibilak organism of DNA genetic material hisabe kaam kore, substance hisabe kaam kore, teneko organism kintu RNA function non-genetic hobo. The function of RNA will be non-genetic in the organism where DNA is genetic material. Okay? In such organisms, DNA produces RNA. DNA produces RNA, and this RNA now produces proteins and forms different types of character, and these proteins are responsible for expression of our trait. আমি actually আমাৰ body যিবিলাক লক্ষণ চাৰিত্ৰিক লক্ষণবিলাক প্ৰকাশ পায়. Suppose I have hair, মোৰ হেয়াৰৰ characterটো suppose black I am speaking now, মই ধৰিছো মোৰ hair colorটো black. এই black hair হোৱা কাৰণটো হ'ল মেলানিন, ই protein এটা হয়. মোৰ হেয়াৰৰ শেপটো suppose straight বা curly, straight হেয়াৰৰ শেপটো ডিটাৰমাইন কৰিছে কিন্তু কেৰাটিন নামৰ আন এটা প্ৰটিনৰ দ্বাৰা. তাৰমানে আমাৰ যিবিলাক কেৰেক্টাৰ বাহিৰৰ ফালৰ পৰা এক্সপ্ৰেছ হয়, মেনিফেষ্ট হয়, the characters we express in our body, they are actually due to protein. কিন্তু প্ৰটিনটো ফৰ্ম আল্টিমেটলি হ'ব জিনৰ কাৰণে, কিন্তু জিনে ডাইৰেক্টলি প্ৰটিন ফৰ্ম নকৰে, জিনে RNA ফৰ্ম কৰি প্ৰটিন ফৰ্ম কৰে. গতিকে there is a relationship found between DNA to RNA, RNA to protein molecule. আৰু সেই যে ইনফৰমেশ্যনটো, this information from DNA is sent ultimately to protein, is called central dogma. এইটো মই ৰাখিবা টাৰ্মটো, এই টাৰ্মটো মই ৰাখিবা. central dogma হ'ল the way in which the genetic materials means DNA transmits or transfers the DNA's character ultimately to express in the form of protein. DNA হিহঁতৰ character বিলাক, DNA property বিলাক, DNA যিটো character, actually আমাৰ body প্ৰতিটো character চৰিত্ৰ প্ৰকাশ পায় DNA ৰ কাৰণে. গতিকে DNA actually code হিচাপে ইনফৰমেশ্যন বিলাক ষ্ট'ৰ হৈ থাকে, নিহিত হৈ থাকে. কিন্তু DNA সেই character বিলাক এক্সপ্ৰেছ কৰিবলৈ যাওঁতে through formation of RNA control করে. মানে DNA synthesizes RNA in order to produce protein molecules. And those proteins are responsible for description or determination of our character, manifestation of our character. গতিকে কিন্তু এতিয়া চোৱা, যেহেতু RNA single stranded হয়, মই কোনোবা পাৰ্ট কৈছিলোঁ, RNA যেহেতু single stranded হয়, গতিকে DNA দুডাল ষ্ট্ৰেণ্ডৰ ভিতৰত যিকোনো এডাল ষ্ট্ৰেণ্ডেহে RNA ফৰ্ম কৰিব. Means out of two strands of the DNA molecule, one strand will synthesize the RNA, or that RNA will now synthesize protein. Out of the two strands of the DNA molecule, one strand only involved in the formation of RNA, or that RNA strand will now form the protein molecule. Protein also single stranded molecule. গতিকে দুডাল DNA strandৰ যিডাল strandৰ পৰা RNA ফৰ্ম হয়, সেই ষ্ট্ৰেণ্ডডালক আমি টেম্পলেট ষ্ট্ৰেণ্ড বুলি কওঁ, টেম্পলেট বা এণ্টিচেন্স ষ্ট্ৰেণ্ড বুলি কৈ থাকোঁ. এণ্টিচেন্স ষ্ট্ৰেণ্ড, টেম্পলেট ষ্ট্ৰেণ্ড, এণ্টিচেন্স ষ্ট্ৰেণ্ড একেটা মিনিং, টেম্পলেট আৰু এণ্টিচেন্স একে. খালি মই ৰাখিব এতিয়ালৈ. গতিকে ধৰি লৈছোঁ এইটো DNA molecule এটা পাৰ্টিকুলাৰ পাৰ্ট. Suppose this is a part of the DNA molecule and two chains of the DNA molecules are here. Out of these two chains of the DNA molecule, one will involve in the formation of a RNA. ইয়াৰে এডালৰ পৰা RNA প্ৰডিউচ হ'ব, এই দুডাল ষ্ট্ৰেণ্ডৰ DNA পৰা যিকোনো এডালৰ পৰা RNA প্ৰডিউচ হ'ব. Okay? The strand of the DNA molecule from where an RNA produced is called antisense strand or the template strand. Template আৰু antisense দুইটা কিন্তু synonymous word. Both are synonym. Template and antisense are synonymous words. Template, antisense are synonym. গতিকে কিন্তু আকৌ মন কৰিবা, ধৰি লৈছোঁ মই এইডাল ষ্ট্ৰেণ্ড আৰু যিটো RNA ফৰ্ম হ'ব, এই DNA ডাল ষ্ট্ৰেণ্ডৰ পৰা যিডাল RNA ফৰ্ম হ'ব, this is suppose the RNA strand being synthesized from any of these two strands. আৰু যদি এই RNA ডালৰ ষ্ট্ৰেণ্ডৰ পোলাৰিটিটো, ডাইৰেকশ্যন যদি আমি 5 to 3 ইয়াত লিখোঁ, to make this strand antiparallel, definitely this strand might be involved. তাৰমানে এই ষ্ট্ৰেণ্ডডাল এণ্টিপেৰেলেল যদি হয়, এণ্টিপেৰেলেল যদি বনাব লাগে তেতিয়াহ'লে এই ষ্ট্ৰেণ্ডডাল, কাৰণ 3-5 এই ৰিলেশ্যন গৈছে, এইডাল 5-3 গৈছে. গতিকে সাধাৰণতে এই ৰিলেশ্যনশ্বিপ কিন্তু DNA RNA চবতে মেইনটেইন হয়. গতিকে এই 3-5 DNA strand will synthesize a 5-3 RNA strand. এই 3-5 তাৰমানে 5-3 RNA strandডাল 3-5 DNA পৰা পৰিছে. গতিকে এই দুডাল ষ্ট্ৰেণ্ডৰ ভিতৰত যিডালেই 3-5 হ'ব সেইডালেই হ'ল টেম্পলেট. গতিকে this is the template strand, therefore it is the template strand of this is the template strand of the DNA molecule. Template strand of DNA. It is template strand of DNA. Template strand. Okay? The template strand মানে কি বুজাল কৰে? The strand out of two strands of the DNA molecule where from an RNA is synthesized. DNA দুইডাল ষ্ট্ৰেণ্ডৰ ভিতৰত যিডাল ষ্ট্ৰেণ্ডৰ বা RNA ফৰ্ম হয় সেই ষ্ট্ৰেণ্ডক আমি কি বুলি ক'লো টেম্পলেট ষ্ট্ৰেণ্ড বা এণ্টিচেন্স ষ্ট্ৰেণ্ড বুলি কৈছো মন কৰিবা টেম্পলেট ইজ সিনোনিমাস টু এণ্টিচেন্স ষ্ট্ৰেণ্ড okay আৰু DNA বাকি যিডাল ষ্ট্ৰেণ্ডৰ পৰা the other strand of DNA molecule where from the RNA is not synthesized is known as sense strand or coding strand or non-template strand গতিকে ইডাল ষ্ট্ৰেণ্ড কি হ'ব তাহলে sense strand or coding strand so the template strand is antisense strand template strand is antisense strand template antisense আৰু coding sense strand the sense strand মানে কোনটা কৰে DNA the strand of the DNA molecule from where RNA is not synthesized is called sense strand or coding strand or the strand of the DNA molecule from which RNA is synthesized is called antisense or template strand okay usually the newly synthesized RNA always take place synthesis of a nucleic acid always take place in 5'3" direction গতিকে 3'5" strandal template হ'ব গতিকে তাৰ মানে ৰিলেশনশিপ এনেকুৱা হ'ল a 3'5" template strand synthesizes always a 5'3" new strand of a nucleic acid maybe DNA or RNA whatever be কিন্তু ইয়াত মই RNA synthesis কথা কৈছো গতিকে এটো হ'ল কিন্তু এতিয়া মন কৰিবা এডাল RNA this is RNA strand a part হ'ল DNA this is DNA these two strands are DNA and this strand is RNA a strand হ'ল RNA গতিকে DNA পা RNA হ'বলৈ হলে মন কৰিবা this is DNA this is RNA DNA ইয়াত নিউক্লিওটাইড বিলাক DNA নিউক্লিওটাইড ডিঅক্সিৰাইবোনিউক্লিওটাইড ইয়াৰ নিউক্লিওটাইড বিলাক রাইবোনিউক্লিওটাইড ডিঅক্সিৰাইবোনিউক্লিওটাইডৰ পৰা মানে DNA পৰা যদি RNA ফৰ্ম হয় there is a transformation of nucleotide from deoxyribonucleotides to ribonucleotide must occur transformation hobe lagibo deoxyribonucleotide must be transformed in the form of ribonucleotide good this transformation is carried out by an enzyme known as RNA polymerase more rakiba this enzyme is called RNA polymerase this is the enzyme RNA polymerase is the enzyme responsible for synthesis of a single stranded RNA from a DNA molecule DNA ko RNA synthesis hua control kora regulate kora enzyme to naam hol RNA polymerase kintu etia mon koriba this formation process of DNA motmalko DNA properties or DNA function or nisna hoy gole kora kini even though I am describing here the properties of DNA but this you can utilize these points but this information it can utilize as the functions of the DNA molecule also DNA property or function tomaloke together bohibo para conglomeration in conglomeration you can study gotike yat jodi ma tomalok DNA property koi ase ekri DNA function boli hobo para okay so the strand of the DNA molecule where from an RNA synthesized the strand of the DNA molecule where from an RNA synthesized is known as template or antisense strand. And a template strand's polarity always occur in 3'5" directions. তাৰমানে 3'5" template strandৰ পৰা এটা 5'3" নতুন strand synthesis হয় হেতু RNA হওক বা DNA হওক কিন্তু now I am describing here synthesis of RNA during the functioning of the DNA molecule যেতিয়া DNA কাম কৰিবলৈ যায় RNA form কৰিহে কাম কৰে এই RNA protein form কৰিব আমি এই process টো কৈছো the information flow from DNA to RNA and then to protein molecule is called as central dogma or central dogma exists in every organism wherever there is DNA jote DNA আমি পাওঁ that central dogma নামৰ phenomenon আমি পাম okay or central dogma therefore represent the pathway of expression of the DNA molecule we call as central dogma okay during the expression of during this functioning during this activity or exposure of the function of the DNA molecule RNA polymerase enzyme required whose function is to synthesize RNA on template strand of the DNA molecule gori mo rakhiba RNA polymerase namor enzyme to DNA template strand or pora RNA polymerase namor enzyme to DNA template strand or pora RNA strand produce kore Okay, the polarity of the new strand always 5-3, and therefore, because antipyrality is the rule present in nucleic acids, the template strand must be in 3-5 polarity. 3-5 polar template strand of 5-3 polar RNA strand synthesis हुआ a process to আমি কওঁ transcription. 3-5 DNA template strand পৰা 5-3 RNA form a process to আমি কওঁ, this process we call as transcription. আৰু যিহেতু এই ফাংচনটো RNA polymerase regulate কৰে বা control কৰে, since this function is controlled or regulated by RNA polymerase, we call this process as transcription. এই process to RNA polymerase control কৰে, RNA polymerase আমি transcriptase concept. The process or the phenomenon of synthesis of RNA from DNA template strand is known as transcription. The process naam ki bolo moi, this process or phenomenon is called as transcription. Transcription, the process is, and RNA polymerase, the enzyme is. Transcription is the process and RNA polymerase the enzyme. Okay, RNA polymerase যেহেতু transcription process to control করে, since RNA polymerase controls transcription process, we call sometime RNA polymerase as transcriptase also. আমি RNA polymerase ভিতৰতে transcriptase বুলিও ক'ব বিচাৰো কেতিয়াবা. Sometimes we call it as transcriptase. Tell why RNA polymerase is designated as transcriptase? Because the function of this enzyme is to transcribe RNA on DNA from a DNA strand. So RNA polymerase is transcriptase বুলিও কওঁ, আৰু processটো তেলে transcription. Remember, this is the property number 1, property number 2, and function of also DNA molecule, how DNA works exactly. But here the formation of RNA by DNA is called the heterocatalytic function of DNA also. A function to come DNA heterocatalytic function of DNA বুলি ক'ব বিচাৰো. Heterocatalysis মানে কি তেলে? You remind one thing, later on we find that যেতিয়া DNA কাম কৰে, DNA perform many different types of functions in human body or organism's body. DNA বহুত বিভিন্ন ধৰণৰ কাম কৰে, কিন্তু তাৰ ভিতৰত এটা ফাংচন কি কৰে, DNA RNA produce কৰে, RNA produce কৰি DNA protein form কৰিব পাৰে. DNA can synthesize RNA, and by synthesizing RNA, DNA can synthesize proteins also. Okay, but in addition to this, the DNA can synthesize its own copy as well. DNA কিন্তু RNA form কৰাৰ উপৰিও নিজৰ কপিও নিজে produce কৰিব পাৰে, মানে DNA can multiply itself. RNA can never multiply. DNA নিজে নিজে multiply কৰিব পাৰে, মানে এটা DNA ৰ পৰা সেই DNA টোৱে আৰু বেলেগ এটা DNA কো produce কৰিব পাৰে. তাৰ মানে এটা DNA আছিল আৰু এটা DNA produce কৰিলে দুটা DNA হৈ গৈছিল. But one DNA can multiply by the process, and the process of multiplication of DNA by itself, that means, is called replication of DNA. This transcription or synthesis of RNA from DNA, or DNA from DNA from a process to replication. Actually, replication is equal to duplication. DNA duplication of duplication term to usually use no koru, use replication. DNA duplication is called replication. Formation of DNA on DNA is called replication. Formation of RNA on DNA is called transcription. Kintu DNA pa DNA form hote nucleotide or transformation provision নাই, মানে DNA pa DNA nucleotides synthesizes DNA nucleotides more number. DNA nucleotide dor পৰা আৰু DNA nucleotide form a process to আমি কি বুলি কওঁ replication. ইয়াত কিন্তু DNA polymerase enzyme হয় কৰে. It is not transcription. গতিকে transcription and replication are different. কিন্তু আমি মন কৰিবা replication RNA আমি নাপাওঁ, কাৰণ RNA নিজৰ কপি RNA produce কৰিব নোৱাৰে. RNA has to depend on DNA for its formation of copies. RNA নিজৰ কপি formation হ'বৰ কাৰণে DNA ওপৰত depend কৰিবলগীয়া হয়. গতিকে RNA cannot replicate itself, but DNA can do that. গতিকে সেই ফালৰ পৰা DNA is a superpower. মানে DNA is a independent entity. It is an independent entity which can replicate. Or A to DNA is an important property. Or here karne DNA genetic material hai. Kintu RNA habo noile hetu ta karan. Okay, tila ami paluge ki swa that tumako lagisil sense or antisense strand mane ki. I think difference you might be observing therefore tumake difference to pala senses or antisense strand mane ki. Itu property number 2 of DNA. First property ami koru complementary base pairing mane ki ase. What is complementary base pairing means? He bar tomorrow discuss already koru. Ebar tomorrow koru ma sense or antisense or because sense strand versus anti-sense strand I have described now. Now I will describe third property of the DNA molecule. Our property. That property we call denaturation of DNA. Third property of DNA. Third property, denaturation of DNA. Denaturation of DNA. Denaturation of DNA. Every terminology or these are very, very important from exam points of view. What is mean by denaturation of DNA? Denaturation means ki bujhai? DNA. Okay. The denaturation of DNA, actually DNA double stranded molecule, and the two strands are joined together, as I have already cleared in my some of the earlier class. Kisman class DNA molecule double stranded holo dual strand kitu idal log lagi thake through their basis, by their basis or at their basis. The base bilakor dara DNA molecular adult strand and double strand log lagi pela eta double strand hoy thake. Kintu there are two hydrogen bonds. Bases join together the adjacent strands of a DNA molecule. Kintu by their hydrogen bonds, by their bases through formation of hydrogen bonds. A hydrogen bonds bilakor number A and T mazot dudal koi thake. Mane every AT pairs up by two hydrogen bonds, and every GC pairs up by three numbers of hydrogen bonds. Okay? There are two hydrogen bonds between A and T, and there are three hydrogen bonds between G and C. So by these hydrogen bonds, the adjacent the two polarized strands of the DNA molecules are joined together. By hydrogen bonds, the two strands of the DNA molecules are joined together. Kintu hydrogen bond desoa jetia DNA kaam koribole jabo. Kintu kaam koribole joa agota prior to the involvement of functioning, DNA molecule has to undergo separation of the two strands of the DNA molecule. DNA kaam kora thik age dual strand separate hobo lagibo. The separation of the two strands of the DNA molecule is actually known as denaturation, or denaturation therefore required to perform the function by DNA. DNA tar kaam koribole hole DNA dual strand separate hobo lagibo. Buji paisa ki jani? I think you are following about this. The two strands must be separated from each other in order to involve in functioning by DNA molecule. Or this separation is possible by dissolution of the hydrogen bonds occurring between their complementary bases. Kikolo? The two strands of the DNA molecule must need to separate from each other. Must be separated from each other so that the single strand of the DNA molecule now produced can involve in functioning. Ite ma agora koisu tumalok dual DNA strand ba jekino adal par ana porisoba pare. Kintu he kaam to korbole holo dual strand ekare thakile no, but the two strands must be separated together. Ite ma agora koisu swa. Suppose it is the DNA molecule. Let me to assume the DNA molecule again like this is a DNA molecule. Itu apu DNA draw korisu. Here say A T G C A T G C. Suppose A T G C A T G C. Here we go T A C G T A C G T A C G. Ebla hol DNA base. AT mazot dual hydrogen bond thake. Between A and T there are two hydrogen bonds present between the two strands of the DNA molecule. AT mazot dual hydrogen bond. GC mazot tin dal ko hydrogen bond thake. GC mazot tin dal ko hydrogen bond thake. AT mazot dual, or GC mazot tin dal ko hydrogen bond thake. Yes, because these hydrogen bonds held the two strands of the DNA molecule together. A hydrogen bond gita dual strand dhori rakhe. Okay, but DNA come arambo habolo jabo, when a DNA is going to work, the two strands must be separated. Kilo separation only possible by dissolution of the hydrogen bonds. Jetia hydrogen bond gita aturi jay, when these are dissolved, when these are the hydrogen bonds, these are H bonds holding the two strands together, holding the two strands of DNA together. Holding the two strands together. Dual strand of globogai rakha factor gita kun hydrogen bonds. Gotike kintu ihato DNA kamor agote ihe dual strand separate hobo lagibo. Because separation can be carried out by dissolving the hydrogen bonds. Dissolving H bonds occurring between complementary bases can cause separation of the two strands from each other, adult strand video separate. Suppose adult strand here separate. Adult strand separate. These are two strands. Adult strand separate. We keep looking. A T Z C A T Z C. So here A T Z C A T 1 2 3 4. Here it whole A T Z C A T Z C. If I love T A C Z T A C Z. It does not follow this. It is single strand because DNA produce a process to whole denaturation. A process to this process we call as denaturation. Denaturation. Okay. So denaturation is the process of separation of the two strands of the DNA molecule, means a double-stranded DNA molecule, by dissolving their hydrogen bonds. By dissolving their hydrogen bonds. But hydrogen bonds can be easily by any dissolve another. Something must be there that can dissolve the hydrogen bonds. The hydrogen bond dissolve kora agent bilak, maybe physical agent, maybe chemical agent, whoever is responsible. Denaturing agent বুলি কম. Any external factor responsible for dissolving the hydrogen bonds known as a denaturing agent. Denaturing agent. Denaturing agent. Denaturing agent. Denaturing agent মানে কাক বলে? Denaturing agent any entity that is capable of dissolving the hydrogen bonds holding the two strands together is called a denaturing agent. Okay, so the process is called denaturation. The chemical or the physical substances responsible for separation of the two strands of the DNA molecule is known as denaturing agent. Okay, a denaturing agent be like this. Denaturing agents may be temperature, this may be temperature, or this may be some chemical agents like strong salt solution, or maybe acids, or maybe alkali or bases, or maybe enzymes, or maybe enzymes. These are temperature, strong salt solution, acids, bases, and enzymes. These are certain examples of denaturing agents. That means they are capable of removal of the hydrogen bonds, and the two strands of the DNA molecules can be made therefore separated. Okay? Here we also alkali, acid, salt solution, temperature. These are actually in vitro agents called. In vitro. Mane ইহঁতে আমাৰ বডি ভিতৰত নাথাকে, আমাৰ বডি ভিতৰত আমাৰ লিভিং সেলৰ ভিতৰত লিভিং অৰ্গানিজমৰ ভিতৰত যদি ডিএনএ ষ্ট্ৰেণ্ডৰ সেপাৰেট কৰিবলগীয়া হয় তেতিয়াহ'লে কিছুমান এনজাইমৰ দ্বাৰাহে হয়. Example of enzyme responsible for denaturation is the helicase enzyme. Helicase is an enzyme. Helicase is one example. Topoisomerase is another example. Single stranded binding protein will find their names later on. Pisat pamge. Kini mora kipa ketaman tam use korisu. Enzymes as well as chemicals or physical agents. Maybe ketiba rays bilak, physical ray, radiations of different type, high temperature, UV rays, IR, NMR, etc. are some rays. They can also dissolve the hydrogen bonds. Okay, anything else, any entity capable of dissolving the hydrogen bonds in order to separate the two strands of the DNA molecule is known as denaturing agent. Okay, alkali, acid, salts are temperature, but radiations of different types, UV radiation over pare. Jar dara hydrogen bond remove kora hoy kintu hebilak bahiro agent body bhitor kam kobo nora ihate. They are otherwise kill the organism or cell. Gotike ihata kekana koy in vitro agents, denaturing, in vitro denaturing. Kintu enzymes are in vivo denaturing agents called in vivo, INVIVO. In vivo agent or ihata in vitro agents, alkali or temperature in vitro. You have body by my laboratory. I mean, suppose we want to separate two strands of the DNA molecule in laboratory conditions outside a living being, then we use any one of these, maybe all of this. But our body ভিতৰত